So perhaps in the interest of time, I'll start. I'll get started. So let me share my screen, and people can. Oh, okay. Hello. Sorry. Some.
Hi, Max.
Still figuring this out. I haven't done much hosting. How are you folks?
Hey, Max.
Hi, Alan.
Hello, Max. How you doing?
Pretty good, pretty good. I figured start a little bit easy, but I want to jump into it, and then people can sort of file in.
Okay.
Hopefully, I'm not holding anybody more up. So yeah, it looks like everybody is invited for the moment. So let me jump into it. Let me start this presentation. So can you folks see my screen?
Yes.
Yes.
All right, great. So I wanted to tell you about our mission and also some kind of state-of-the-art technologies. So, okay, here we go. So this is meant to try to provide practical value, and these are the questions that I'd like to give you answers before you walk away. What is the state of the art? And I've added diagnostics briefly as well to drug development because I've spent a lot of time and certainly feel very strongly about it. And then I'll talk about the better medicine, which is precision and personalized and outcome-based tools for it, the role of biomarkers in general and in COVID, and also touch upon kind of this conceptual framework of so-called biocomputation. But it's a little bit disobedient. So, so first of all, briefly about me. Some of you know me, some of you don't. I've been, it's about 20 years since I finished my undergraduate, have done graduate work in Whiteside's laboratory with microfluidics and biochemistry, biophysics. And then did, as you can see, some work in drug discovery, a postdoc, and primarily was working on diagnostics and companion diagnostics using microfluidic techniques. Hello, can you please mute? Thanks. If you have questions, by all means, interrupt or raise hand, even better. I'll try to address them if they're appropriate for the moment. Otherwise, I'll kind of try to walk through what I have, which is already a fair amount, and then try to get to the questions. So, so this is what I wanted to do from the beginning and how I see kind of my role. It's trying to connect the means to the ends. The ends are to provide improved healthcare. And the means are, as I will tell you about, not just analytical, which is actually where I started kind of as a bit of a math whiz, but specifically in terms of hardware. How do we actually manipulate the biology to perform meaningful measurements? What does it look like? So, you've already hopefully seen this mentioned in the invite email. So this is kind of the structure that I'll be going through. So first, let me briefly talk about the hardware for diagnostics. And this is kind of actually a step back to think about hardware for tools in general. So there's been a confluence of three big things that have been producing a lot of benefits for us, starting from material science, computation, and now biological. And they are feeding into one another. But there is a problem with this flow. The relative speed of biology is nowhere near the speed of computation. And so this biological computation where you take a sample, add some reagents to it, and get an answer is very slow. Certainly in comparison to the challenge at hand. This is a yeast network. Human network has about 100,000 proteins. There are so many connections we are only scratching the surface simply based on the number of experiments we're able to conduct. And then, of course, there's the quality of the experiments. So one of the points, and this is, still the case, these slides actually I took from presentations more years back that I, care to mention. But the difference in speed is in the kind of, 10 to the 12 range. It is enormous, the deficiency of our, tools for measurement relative to the task at hand to understand this biology and then to be able to affect it. So what does it look like for diagnostics? Science, well, there's supposed to be, this feedback also generating information in the form of biomarkers. And as you can see here, it feeds back. But on the diagnostic side, there is a range of these instruments. And the point of this is that, as we're experiencing with COVID now, there is a need for decentralization. So these central lab analyzers are potent, but not suitable for the task at hand, for example. And these actually should become familiar. These are HCCG is the human gonadotropin, protein, pregnancy hormone. This is a point of care instrument from Abbott called iStat that does electrochemistry. The challenge that these instruments are facing is that the FDA doesn't discount their form factor anymore. They have the same requirements essentially as for central lab, which is made adding new items of menu to these instruments that have been approved a huge challenge for the company. What's coming is miniaturization, microfluidics. And it's going to take this concept. This is something from a competitor of Abbott, Roche, bunch of cobas systems strung together, very, very large system, and I'm sure they're very proud. But I kind of think that this has limitations. This size, it has its time, and it can rule for a long time. But eventually, small and nimbler things make it more challenging, and that's where microfluidics comes in. So to finish up, diagnostics has made this problem apparent. And although this website with a blog that I posted talks about how we have, not done particularly well. In fact, in many cases, this is in general the worldwide crisis. So for example, here, I'm illustrating, the pregnancy test equivalent of COVID positive and negative, which is exactly the kind of technology that they have now been adopting for serological testing. And so this is the best, kind of technology for point of care distribution of these tests. But these technologies have been around for 40 to 50 years, certainly starting in literature since the 80s. These pregnancy tests have been around. They suffer from false positive and negatives, and to have them as the fallback option for COVID shows the lack of investment and the magnitude of the problem we have with diagnostics. But there are many examples in literature and also personal examples. So this is a very heartfelt point for me. But the technologies related to detection of these infectious diseases, microfluidic technologies, also have relevance for drug discovery. So let me tell you about the processes for drug development. Well, they're very inefficient. So this is the kind of attrition, that's typical of drug development process. You can see how many years this takes with the initial, lead generation, the preclinical, clinical. In oncology, my specialty, historically, the batting average of clinical trials was about 5%. Now it's been elevated to 7% in general. So these are very poor numbers considering the vast investment of millions of dollars and years of research. And that's because the tools that are being used are not exactly right. We have CRISPR, we have some new tools, but the way of handling them, the robotics, are very clunky. So there is this discord. But also there is this issue of use of biomarkers. They've been becoming more prevalent overall, but sadly for COVID, that's not what's happening. And I'll mention about that in the clinical trials. So there needs to be an improvement in the technologies for drug development overall to address the terrible attrition and waste, to accelerate and to deal with the acute problem at hand, for example, COVID. Of course, precision medicine of biomarkers is needed for other areas like cancer. This is just a list of some of the attrition from 2018 when I was doing this initially last year. That was when the data was available, and lots of big names. And the point to see from this kind of summary of it is look at how late these failures are occurring from big companies, Janssen, Merck, others you may know. This is a multi-billion-dollar company. Many failures in phase 2, 3, multiple phase 2, 3. So this is millions of dollars, down the drain, lives kind of endangered for, a lack of proper preparation. And recently, an MIT study has established that the cost of development of a new drug, a high-quality blockbuster drug, is $2.6 billion. And we are paying for that inefficiency in this fashion. So what's happening is, advent of more relevant biology, and it's a tsunami change for pharmaceuticals. They're trying to adapt. So what it looks like is, we're changing from the traditional flat monocultures that do not have good translational value to 3D cultures and microfluidics. So this is actually, these are literally the names of a couple conferences next week that I'll be kind of summarizing. But this in large part is geared towards improvement of biological relevance, giving it more physiological relevance. And so these three-dimensional cultures have improved microenvironment, cell-to-cell contacts, but in this middle panel, they're still in some kind of a static gel-like media. So what is needed is, of course, fluidics. In the body, without blood circulation, the body passes away. And there are tools that are starting to address this, to provide this precise and automated control. They're called microphysiological systems for the goal that they address. And this, as I've mentioned, is suitable for a number of applications for precision medicine, not just in COVID, in cancer and other areas. So the way these tools look like, just to give a quick idea, it is a combination of things that provide this kind of, this is a somewhat futuristic rendition, but you can see artificial channels, three-dimensional growth of biology, and ability to perfuse drugs and to monitor this. So there are three key components that come to in play here. Two parts from microfluidics, the hardware pumps and the consumables. And from biology, these improved three-dimensional constructs such as an organoid. The one here is a stained organoid that's showing blue nuclei in blue with DAPI staining, and two other colors, which kind of reveal some of the complexity of this system. So that captures minimum relevancy of an organ. So that's why they're called organoids. So they are also, as I mentioned, highly suitable for cancer. So these organoids can be used for isolated and screened for various drugs. They are, often for study purposes taken from fatty adipose tissue and reprogrammed with various factors to generate a number of tissue types. And these were the ones that were known, some 6 years ago. The list has grown, not tremendously. And they have a bright future, and largely intestinal and gut organoids seem to have the most kind of promise, but there is a lot of need for neurology. So brain on the chip and other kind of things are also, coming. These have a number of validations in clinical trials. Well, actually, I should say preclinical trials have been completed. Clinical trials are ongoing for these systems. There's certainly a lot of publications, and these are all in kind of my area of cancer, by, again, organoid type. So these are actually kind of tumoroids. So this is validated. Next, I want to jump right into the next step. What does precision and outcomes-based medicine look like? Well, first of all, it generates biomarkers of different types. There is also cells, which are not shown here. But this is my list from a website of biomarkers for COVID-19 taken from a number of publications. And the point to observe here is that they come in a few categories. There are cells, here the immune type cells, inflammatory proteins, proteins involved in blood coagulation, which is one of the hallmarks of the immunopathology that's inducing this microclotting. And then small molecules, metabolism has strong comorbidities for COVID-19. So this is what's an example of the repertoire of biomarkers for precision medicine. So that's what is required. We require that type of data. But what does it give us? Well, we take a bunch of first of all, we can better detect. So that's the diagnostics part. But I'm focusing now a little bit more on treatment. Especially for COVID-19, since these trials are ongoing, this is what's missing by and large, trying to differentiate patients by biomarkers. And for that, they need to be measuring them. And by correlating these biological entities with response, they can identify, the likely responders. And prospectively, then, they can predict this for future patients. So first, this type of relationship of biomarker validity is established in trials, and then it is used in ongoing treatments, and it actually could be, continually refined. But the point is, there is this now designation of drugs for specific groups of patients, and it's based on cohort behavior, response. People that have similarities to you in the number of biomarkers that identify that cellular state. There are actually a couple types of precision medicine. One is prevalent, and I've worked on this for hematological cancers. And I want to contrast that with the better form of, complementary diagnostic versus companion. Companion diagnostics are sponsored by drug companies. So they're looking to find the right patients for the drug. And literally this is what it looks like. So if you're not the right patient, sorry, we can't help you. This is what it looks like in, the clinical trial. And then afterwards, once this trial is approved, they may still, get reimbursed. But a complementary diagnostic is really patient-centric. It's trying to analyze the, repertoire of treatments for a condition. And also, this is more easily connected to outcome-based medicine. So, flat, where the insurance company pays for the treatment regardless of how well or poorly it works, versus incentive-driven. So this is obviously the state to strive for. And there are technical hurdles as well as kind of economic hurdles for regulatory purposes. But this is sort of simplistically what it looks like. Right now, once the drug is approved, we're paying for it regardless of outcome. We need to incentivize the response curve. So if good outcomes are achieved, companies are reimbursed on the grade scale or some kind of a Gaussian curve. So in this way, precision medicine is connected to outcomes-based medicine. So that's the point of that. Now, I'm taking note of some questions, but let me just continue into the tools for this. As I was saying, specifically focusing on drug development. So right now, there is a bit of an artificial separation between drug development and development of these biomarkers for companion diagnostics. The oversight is also divided between departments, but they are actually encouraging co-development to erase that artificial boundary. And this will presumably accelerate the whole improvement by helping with this translational value, these arrows jumping from preclinical models to human, and then getting approval and efficacy. So there is a way to aid this. We provide better tools for biomarker development at the level of microphysiological systems, and then these tools for companion diagnostics and precision medicine to measure these biomarkers. That provides connectivity and overlap between the preclinical and the clinical. It passes the baton by the biomarker, as opposed to moving from 2D monoculture to a mouse model to a human, with big gaps, and it results in a great inefficiency that we all pay for. So there's a lot of things that need to change, but we're working primarily on the hardware front, obviously, to enable these specific areas with the focus on biomarkers and also analytics. So what do these tools look like? Right now, the organoids are grown in vats. So this is what companies like Celeste do. And then they are actually having to screen them in systems, including microfluidics, to have some kind of a chance to have consistent response. This is one of the problems I've heard from a number of pharmaceutical companies that are trying to use organoids right now. It's a very poor system for screening because of heterogeneity of the starting material. Also, there is an issue with how the screening process works. They're using a very expensive and heterogeneous matrigel, which is derived from mouse sarcoma cells. It has unpredictable regulatory factors that affect organoid development, which leads to some exacerbation of heterogeneity. So this is what some of the screening can look like, for example, for immuno-oncology applications. But then there is this very deep piece of gel that needs to be sliced with confocal microscopy, and that leads to a lot of waste. So this is something that can be improved using microfluidics, and that generates efficiency. And of course, there is the benefit of the flow itself. So not only is microfluidics capable of capturing more organoids, it's also delivering flow and this more efficacious kind of overhead space for these organoids. So here you see kind of a microfluidic scheme for trapping organoids. And the panel on the right here shows a treatment. So blue, as I said before, is typically DAPI staining, so it shows that the organoids and nuclei are still there. And green shows a specific biomarker which is suppressed due to treatment. So this is a time kind of a response where all of these organoids are undergoing the same treatments. But if you isolate them in wells, you can do multiplex treatment and have improved screening conditions. So I've mentioned application for immuno-oncology. So here's kind of an example of T cells engaging and killing a tumoroid. So the T cells are in green, they mob the organoid, which actually loses some of its protein staining and kind of deflates. But importantly, as you can see here in the bottom, there's a couple of important endpoints. Tumoroid volume is not the only differentiator here. There is another one based on engagement of the T cells. This requires observation, and as I mentioned, microfluidics can help with fluorescence confocal imaging and delivery of reagents and other cells. So working kind of backwards from the goals, as I said, connecting means to the ends. If our goal is precision medicine, which enables this identification of proper cohorts, we need biomarkers that will help to define disease and then the state of disease. And there are a number of categories that are necessary for measurement. The tools that exist are expensive, clunky, not portable. They include techniques like flow cytometry, electrochemistry for metabolites, but it's also possible to do protein-based assays, optics for cells. And these are the applications. So the coloring that I'm pointing out here is my kind of own approach and contribution. I have worked lots in diagnostic, but it has regulatory burden. So the starting points are kind of indicated by the color, the intensity of the greenness. The research microphysiological systems, they're called RUO, research use only. So they're essentially as long as pharmaceutical companies are interested in them or contract research organizations, the FDA will not regulate. Clinical applications have obviously regulations, and diagnostics have even longer development cycle. But microfluidic tools can help all of that. And the goal is to provide these better measurements for diagnostics and drug development. And hopefully you can see why microfluidics is kind of a key technology that combines these. So I've mentioned microfluidics and how it works. There are all these improvements that are possible. So these are the benefits. And I wanted to now give you a few examples of the systems that enable this, just to kind of give you a sense of what they are. First, let me go over the problem that exists. The organoids, which you can see here, develop a dead core, a necrotic core. In fact, it's not completely resolved with fluidics, it's ameliorated with fluidics. But there are ways to build on this. So this paper from Linda Griffith, who's one of the luminaries of this field at MIT, shows some of the benefits of microfluidics, but it needs to be built upon. And I'll mention right now a model that illustrates this kind of very clearly. So even if we step away from the full complexity of the perfusion of oxygen and hemoglobin, which is got consumption in the reaction, the diffusion, and the convection in the in the veins. This is a complex problem, but in fact, to demonstrate it, one doesn't have to go to all this complexity. We can simply analyze what happens to a tissue which is not vascularized, not perfused. So we'll consider reaction diffusion only when the outside is perfectly oxygenated. So we're giving it sort of maximum oxygen that's observed. And this model, which, okay, there it goes, is showing this necrotic core in white. So oxygen gradient is shown in this rainbow color. But as you can see, it starts with reduction of oxygen, hypoxia, and then it progresses to death and necrosis. So obviously, past a certain critical size, these organoids have dead tissue, makes them very difficult to use. This is a problem that's not fully realized. There are a number of solutions that are coming into vogue. Microprinted capillaries, all kinds of ways to provide this actual movement of fluid, not just diffusion. But biological vascularization is best. So I've hopefully already pointed all of this out, how this concept is an important driver to advance this kind of state of the art. So now, so yes, just to kind of reiterate, the existing multi-well plates are static, and the fluidics that are coming are suffering from kind of low multiplexing level and low sophistication of the flow. So this is what we are working on. But in the meantime, there are some solutions that are available to kind of deliver these advantages for especially preclinical research, where these better screening systems can be used right now. So one of the roles that FlowCell plays is to distribute these systems that exist while we're developing our own. So there are a number of systems that are all dependent on microfluidics, from 3D cell printers to dispensers to sorting. Actually, this is a 3D cell printer. To microphysiological systems, kind of the core technology that I've been mentioning. So I wanted to give you a few examples, just very briefly, with the disclosure that some of them are being distributed, others are not. So this is not any kind of a push. So this is a very interesting system with a reciprocal pump, which drives this perfluorinated alkane, which functions as a liquid blood. It has significantly better ability for oxygen diffusion, and it's able to actuate this liquid with isolation in wells. So it's a very clever system to keep kind of delivering oxygen to organoids that live in these frits. There are other systems that are suitable for observing larger pieces of tissue, also with membrane systems. There are interesting solutions with nanomaterials, which simulate what happens in the body. And these can be made as essentially porous mats into which one adds cells. And there are a number of applications that have already been validated in oncology and neurology primarily, from breast cancer to immuno-oncology. There are microprinted cages. These cages have the added feature that there is some ability for liquid to actually move through the organoid. As I was showing, it's critically important to be able to actively move the fluid through the core of the organoid. Otherwise, it dies. And we've shown critical sizes that are consistent with literature values where tissue outside of 130 microns away from vasculature becomes hypoxic, and then it will go actually necrotic. So other solutions that enable organ on a chip or some organoid look like this. They are, as I've mentioned, a little bit clunky. They have very large pumps, small chips, and are generally expensive and silo-like. So it's an evolving field, and certainly the products need to get there. This is one of the companies that has made a good choice in multiplexing and using a workflow that is suitable for pharmaceutical companies. This is a 96-well plate. This is the underside of it, and it has microfluidic channels on the bottom that allow establishment of a gel with, for example, neurons and extracellular matrix with a fluidic channel where medium can be perfused. And this can be used in a number of applications, but the fluidics is rather limited. The fluid actuation, as you can see shown schematically here, is driven by gravity in these pretty large channels. They're actually mesofluidic, not millifluidic. They need to be about greater than 200 microns in width to be able to move fluid on a rocker. So just a quick point about other microfluidic products that are of innovative value that are non-microphysiological systems. So here's a very interesting cell gating system. It's related to flow cytometry from Molteni. They typically use absorbent phase, which is what their logo illustrates, but here they have a mechanical valve, which is perhaps not the best solution, but they have a very elegant integration of it at the bottom of a flask. So this flask fits within the instrument, and it literally gates cells based on staining, and it will take white blood cells on one side and isolate based on biomarkers, particular population. There are other tools that are notable, such as Illumina, of course, that has broke Moore's law even for the price of sequencing while putting money in their own pocket. And this is, their kind of lineup. And, after a while, they hit, a kind of a bare bottom. But the fluidics is actually fairly primitive. The greater part comes from microarrays, which is a technique I've also worked on for several years. The microarrays are printed on the bottom of the channel, and there is sequencing by synthesis that happens within these channels. So a very good business. 10x Genomics, another sequencing solution focused on single cells. So their microfluidic solution is a very simple encapsulation of a cell and a bead that encodes, that cell type by a genetic material. It generates an assembly of these droplets as individual cell microreactors, and then there are PCR reactions, and they produce a pool of DNA, but there is encoding of the DNA from the cell by the bead. So it's a clever solution, but as you can see, the fluidics that enabled that revolution is very simple. But again, it's at the core of it. Here's another very exciting tool from Berkeley Lights. These are expensive instruments. This is their initial flagship. As you can see, it's a walk up to instrument for a couple million beacon. They made a benchtop instrument for a mere $1 million, but the consumables are still very expensive. They're about $10,000 a piece. And what they do is they trap up to 14,000 cells individually using optical tweezering. So this is what they are showing here, but it's a very slow process. The loading takes about 30 minutes. And there are better solutions to achieve this. But the price point signifies how relevant it is. They've sold about $150 million to a unicorn company called Ginkgo Bioworks here in Boston. And also one of the applications, in addition to synthetic biology, is development of monoclonal antibodies for COVID. So they've been partnering, and they've announced that they are accelerating this. So we'll see what they come up with. So my idea of biocomputation is, that there are these layers, but first there need to be tools to measure these for the measurements. From them, one can identify validated biomarkers, and those can be utilized. This is actionable kind of scale, predictive assays, biomarkers that correlate with patients who respond to particular drugs. And this, as I mentioned, a little bit drug-centric, but if you put several of these together, it can be made patient-centric. So this is what we're trying to develop, but this idea for biocomputation is strongly wedded to the necessity for physical measurements of these, not just analytics, but having quality information. So now jumping into some specifics for biomarkers in COVID, which is kind of an acute application. So one of the things I've done is analyze US clinical trials for COVID by mechanism of action. It exists on a website called usclinicaltrials.org. And these are graphically clickable, so you can navigate into subcategories of immunosuppressants or antivirals to see, for example, what other analogs there exist to remdesivir. Actually, what's interesting is not only what exists as analogs, because remdesivir is a nucleotide mimic, but what generic drugs in that category are not being tested. But this shows where the effort is going by this grouping of mechanism of action. In clinicaltrials.gov, you can find all of these, but they're just not organized. They're just a big pool. So this is a reminder of some specific cells. These are all T cells. So COVID plays rope-a-dope with the immune system. It's an immunopathology. It suppresses the innate immune system, but stimulates the adaptive. So that leads to T cell exhaustion. And also depletion of natural killer cells. So this can be seen in neutropenia, depletion of neutrophils. There is also hyperinflammation. This is specifically bad in the case of cytokine storm and interleukins, like interleukin-6, the gators there. But they are rather late predictors. Earlier predictors fall into the acute phase proteins, like C-reactive proteins for calcitonin, but there are completely different biomarkers having to do with coagulation, which impact people differently. And also, as I mentioned, metabolism is hugely important. Diabetes is one of the largest risk factors, as are some of these enzymes and levels of lactate. So these are all important pieces of information to predict the likely progression of disease, but also they need to be present during treatment. And right now, by and large, unfortunately, it's not happening. There are two risks that you perhaps haven't heard about. One is called antibody-dependent enhancement, and the other one is enhanced respiratory disease. And these are both bad types of enhancement. And this paper here shows what they are. The antibody-dependent enhancement is infection of otherwise immune otherwise non-targeted immune cells through the back door. They have antibodies by this FC receptor, which holds the antibody by the tail. But this can become a back door for subneutralizing antibodies. This has been seen in vitro a lot in the dish. So there are lots of publications that talk about this, but the CDC in its guidance is not even talking about this. They have finally started to address the prevalent immunopathology, which according to what I've seen is present in at least 85 cases of animal studies, as well as human trials for SARS-1 and the predecessor MERS, which are very closely related to COVID, where the cytokine storm, the enhanced inflammation, leads to enhanced disease. And this is most commonly manifest by invasion of the lung tissue by immune cells, and desquamation of cells by production of nets. These are neutrophil extracellular traps. They literally DNA strands that produce these nets. It's both an acronym and literally a net that tries to reduce traffic. But the immune system does great damage. That's why a lot of the treatments nowadays still are immunosuppressants, or even immunosuppressants are the strongest of this category. But again, they're not often used in the clinical trials with treatments. So talking about the cytokine production, there's a couple of alternatives. Sometimes the infection basically goes away with the cells, as I've mentioned, and the cytokines. Here, with the adaptive response relaxing and here there is a persistent activation of cytokines. So this is this relapse of patients who have started to get better at days 7 to 9, and people can perish here. And I have a personal friend who is 22 who passed out at a doctor's office. It's a good thing, we kind of got him ready. And what's needed is diagnostics. So, for example, this is showing a meta-analysis of one of the acute phase proteins, procalcitonin, from several studies deriving a consensus. So this is how biomarkers should be established. But again, unfortunately, we're not there yet. So speaking of guidance, this is kind of pretty severe. Just recently released guidance they talked about efficacy of at least 30%. But then looking a little bit deeper, this alpha adjusted confidence with 30% cutoff talks about the true kind of threshold. So alpha adjusted is statistically adjusted by the confidence interval. So in fact, this is the true placebo-controlled, statistical kind of efficacy where they're placing the threshold for drug approval. And there are other things where there are numbers, that it kind of takes a while to even understand why they would have, a primary efficacy endpoint set at greater than -10. The point is that they have very low expectations, and they're only talking about one of the risk factors I've mentioned, this enhanced respiratory disease. The antibody-dependent one is not even mentioned. Granted, it's been only seen in the dish, but it's been seen in a lot of contexts in very relevant human tissue in organoids. So this is clearly a risk factor, and they're not actually advised to look for it yet. So in summary, there are kind of two paths. What we're trying to motivate by our tools and this information sharing is biocomputation that's driven by measurements. And for that, we need tools that will give us this precision data for defining the state of disease using biomarkers. Then AI can be used to analyze behavior and correlate, response to biomarkers and, achieve response in all kinds of subgroups of population. But without better data, biocomputation that's driven by AI simply and doesn't have the quantity or the quality of data is kind of in the garbage in, garbage out paradigm, and will result in efficacy of, again, maybe around 30 to 40%, which is common for cancer single agent, stuff before biomarkers. I should mention that in that field, because I've worked on companion diagnostics, I know from literature that companion biomarkers for cancers have more than tripled drug approval rate. And so it's good for companies, but it's also obviously resulting in better outcomes. But right now, we don't have the right incentive yet. So that's what we need. So that's why I'm sharing to popularize the need to fight for precision medicine at the level of technology, but also a regulatory and reimbursement level. So that's basically the end of the presentation. So now I can take some questions. So you guys want to chime in?
I did want to clarify something actually. In my model, the pretty rainbow animation, right? It's not actually an animation of a growing organoid. It is every single frame. I probably shouldn't have made it look so much like one organoid, is a different organoid and calculating the dead zone inside that one organoid. So it's not a dynamic process. That's all. Every single one is steady state. I probably should have made it more obvious.
No. Thanks for that, Henry. I know, again, this distinction is probably a little bit, a little bit technical. But the point is, at steady state for an organoid, if you magically, materialize this chunk of tissue, there is a certain core that will be so hypoxic it will die by necrosis.
Yeah.
Yep. Thanks. So did anybody have more questions? So there was a question about website for clinical trials by Abby. So it's called usclinicaltrials.gov. I'm gonna share it with everybody. Actually, I'm sorry, .org. The government website is clinicaltrials.gov, which is housing over 250,000 clinical trials, and they're not just from US, they're actually from the world, lots of Chinese trials, because they want to have the opportunity to share in this market. So these are kind of a very large collection, but I've tried to organize them. So other questions? So, well, if you guys don't have any more questions, I'm having a little bit of connectivity issues, then you can recover the last few minutes. Next week, I'm going to be attending two conferences, one on microfluidics and biology, the other one on organoids, transition from 2D to 3D/organoid, and then that'll be Monday to Tuesday, and then Wednesday to Thursday. So on Friday, I'll have another summary webinar.
Okay. Hi, Max. Can
Hi.
you hear me?
Yes.
Okay. Thanks for the updating. Interesting.
Yep.
You did a lot of work on COVID-19, yeah?
Yes. Right now, just at the level of reading literature and connecting with some validated applications. So some of the companies that I've mentioned, they have applied their microphysiological systems to cytokines in general, but also to COVID-19 specifically, because it's such a central issue.
Yeah. Good to know.
Mm-hmm. Thanks. So, Abby, to your question about cancer organoids, they are actually, if they are, you're absolutely right. If they're derived from a patient, they could be called a spheroid, but because cancers have a kind of neoplasmic qualities, right? That's one of the paradigm defining characteristics. They're able to proliferate. Ooh, tongue twister. Cancer organoids are distinguished from spheroids by having stem cells. So as long as this regenerative aspect to it, you can call it an organoid or a tumoroid when it comes from a patient. I would say in both cases, I think spheroid is a little bit more for simpler 3D clumps that don't have that regenerative capacity. A question from Vitaly about prototypes. So there are only models such as the ones you and Henry are working on. There are collaborators with whom I'm going to be developing these tools going forward. So they have some experience. I wouldn't say prototypes just yet. It's kind of at the level of components at the moment. So we'll be integrating and improving pieces. So let's see what else. Sorry if I've spoken quickly. I've tried to get through.
Max, yes. Sorry, it's me, Vitaly. I have a question about verification. How we are going to do this verification process to make it like 100% sure that it works just like we are supposed to? Do you have some kind of ideas how we should verify our models or so?
We'll make prototypes. There are actually 3D printers now that are capable of printing microfluidic channels. So we can do that. There are opportunities to do photolithographic, or embossing kinds of prototypes. So the difficulty is that mass production via molding is difficult to connect with prototyping. And what I'm giving you are examples of things that would allow us to prototype. So by a number of these mechanisms, whichever is easiest, would make prototypes and test.
But 3D printing won't allow us to print that precision that we want to have.
Well, that's exactly why I'm hedging, and it would require kind of thinking it through. There are issues with resolution of embossing, and photolithography has perhaps the best. So one would kind of need to consider, that's why, of the sizes and resolution. It depends on the size of the organoid one's trying to trap, the challenge. Since we're not doing single cell perhaps trapping much larger organoids that are grown in batches, I think we could get away with simpler prototyping techniques like embossing. And there are materials and tools that are quite tractable for that. But the bottom line is we would absolutely do prototypes. And then after we prototype, we would probably get some in the hands of some early adopter customers before we get too excited.
Okay, thanks.
Sure. So, well, thanks everybody for joining. Just a couple minutes left today. So if I haven't gotten to your question, please email me. But as I said, I'm very happy that you guys were able to make the time and listen in, because it will help to set the kind of ground up for some future communications, which I will want to make of practical value, including next week the review of the conferences that will be taking place here in Boston. They were delayed from March. But it's perhaps they've even advanced the ball in that time. So thank you, everybody.
Thank you.
Bye now.
Bye.